In. So let's begin. So I'm happy to introduce Professor Euler as our guest speaker in the Bar-Ilan Vision Seminar. Dr. Euler is a professor in the Center of Integrative Neuroscience, and is the co-director of the Institute of Ophthalmic Research, University of Tübingen in Germany. He graduated for his PhD in neurobiology with magna cum laude in Max Planck Institute for Brain Research under the supervision of Professor Wassel and Professor Normeyer. His first postdoctoral fellowship was with Professor Wassel and then with Professor Masland at Harvard Medical School at Boston. He received numerous awards for his excellence in science and is actively engaged in many scientific committees and activities and review boards. He published numerous publications in high-impact journals, where his research is focused on understanding the retinal basis for vision in several species while using high-end imaging and electrophysiology technologies. Current talk title is Natural Visual Stimuli for Mice, Reloaded. So let's welcome Professor Euler, and we can all start. Please.
Yeah, thank you very much for the kind introduction and for the invitation to give a seminar here in this series. So as you already read from my title, I have given a similar talk already last summer. So, but I think we have enough new stuff that I could update it or made a reloaded version of this talk. So my lab is normally concerned with computations in the retina. So we try to understand how visual information is processed by the retina and then handed on to the brain. And in the last couple of years, we got more interested in, or additionally interested in natural stimuli. So in the actual inputs to the retina that the retina receives if an animal is running around the world. And we're doing this specifically in mice. And the work that I present today is part of a collaboration with Laura Busse in Munich, long-term collaborator, but also collaborators in Tübingen here with the labs of Katrin Franke, Philipp Behrens, Matthias Bethke, with Alex Ecker, who's now in Göttingen, and the lab of Fabian Sins. So a multi-lab collaboration. So I think I don't have to repeat this here in the crowd, but we all know that sensory systems and also the visual system is adapted to the environment, to the habitat, to the viewpoint, to the viewing strategy of an animal. And this is illustrated here in a figure from a review paper that Tom Baden and Philipp Behrens and I wrote 2 years ago, where we compare here typical scenes from the environments of primates, mice, hawks, zebrafish, with the ganglion cell density distribution in the retina. So in a primate, primates are scanning their environment, so they have a fovea that is used to look at these scenes. On the other end of the spectrum are, for example, zebrafish, who have this density here in the lower retina, which is implicated in predator avoidance and also in hunting. You can look at hawks, for example. They have developed two foveas. From the first glance, the mouse retina doesn't look very exciting in this comparison, but I come to this a little bit later. So if you want to study vision in an animal, you have to consider these factors. And this is something that is getting, I mean, it has been studied all the time, visual ecology, but there's an increasing interest in studying this again and with modern methods and more systematically. And this is really a very nice development. I just want to highlight two studies from this year. This is a study where they took a virtual environment here and looked at different viewpoints using avatars and looked at the statistics from the different viewpoint and pointed out how the statistic changes if you're a human or a cat. Or there was a very nice study from the Nielsen lab here where they looked at the spectral composition of images from plants from the viewpoint of a bird, for example, here. And what they showed here with these images was that the UV vision in these birds might help birds to detect nutrient-rich parts of plants that are not fruit, or not very not signaling that they can be eaten. So they looked here at these parts of the plants and show that they have specific contrast if you include a UV channel. So these kind of studies are very interesting because they tell you something about the environment that an animal is optimized for. So we study mice, and mice are at the first glance not very interesting. And I think this is partially because we don't understand them. And this was very nicely illustrated in a study last year, which I found extremely exciting. Has nothing to do with vision, but it's a very nice example for what can be hidden, but what you cannot see if you always look from the human standpoint. And this study was about facial expression in mice. So they looked at mice. These are it's the same mouse having different facial expressions, and if you look as a human, you wouldn't see much of a difference. But they trained an AI to recognize the differences, and that this AI could then assign different facial expressions in response to, for example, taste or to fear or so in these mice here. And I'm pretty sure that other mice would recognize these faces. But this shows that we have to sometimes go away from a human standpoint and try to see a bit behind what we're expecting to see. And this is what we are trying to do with mice. And I showed you this example here at the beginning. When you just look at the ganglion cell density in the retina, it doesn't look very exciting. But in the past couple of years, it showed that there are interesting regionalizations in the mouse retina that may be linked to the environment. So things that appear in certain parts of the visual field environment. The first regionalization, and this is the one that I will talk about most in my talk, is this gradient in photoreceptor opsin. So in the upper retina, you have a lot of M opsin, so green sensitive opsin. In the lower retina, the ventral retina, you have a lot of UV sensitive S opsin. And I will show a few details about this later. But on top of this, there's evidence for a couple of ganglion cell types in the mouse to show a similar regionalization. And there's still a lot of debate about whether this is true, what this is for, and so on. The best example for this regionalization is probably the alpha sustained alpha cell, which changes its size depending on where it sits in the retina. So it has a much smaller morphology here in the temporal part of the retina and becomes much larger in the nasal part of the retina. This has been studied using transgenic mouse lines. And if you want to look into this a bit more, I can highly recommend a talk to you by Daniel Kirschensteiner, who was presenting last week his work on mice hunting crickets. And they showed very nicely that this temporal area in the mouse retina, where these cells are particularly small, plays a role in this hunting behavior. And the mice use this area to find crickets, and they use vision to hunt crickets actively. So this shows that there are interesting things also about the mouse retina. And this means that we have to look into the environment. Before we do this, a bit more about this opsin gradient that you can see in the mouse. So mice have two opsins and the rhodopsin, and the S opsin peaks at around 360 nanometers. The M and the rhodopsin peak around 510 nanometers. And this is a difficulty for experimenters, and this is also ties into the point that I made at the beginning. You don't expect an animal not to see a scene on a screen like you do it. Yeah? But if you show a stimulus to a mouse on a normal TFT screen, you won't excite the UV cone, because the TV screen ends with its spectral range exactly where the S cone starts. So when you use a normal TFT screen, you only excite the rods and the M cones. The cones are distributed in a very specific way. So in the upper retina and the dorsal retina, you have the mosaic that you expect from normal mammals. So you have a few S cones and a lot of M cones. And if you go to the ventral retina, the M cones start to co-express the S opsin. So they become more and more UV sensitive. And this is shown here in immunostainings. And this is not something very specific to the mouse. This also is seen in other animals. It's present to a certain degree in rabbits, but also in hyenas, shrews, and many insects have this kind of organization of UV sensitivity across the ventral axis of their visual system. And interestingly, this is also different in different types of different species of mice. So here we have examples for different mouse species. The house mouse, which is probably the closest to our lab mouse here, but also the steppe mouse, the wood mouse, and the pygmy field mouse. The upper two species have this regionalization of the M and the S opsine, so they show this gradient, while the lower two species don't have this. So these species here has no co-expression, but an even normal distribution of S and M opsin, and this mouse here is lacking the S opsin. And from the environments where you typically find them, this kind of points to a purpose of this UV gradient. So these two mice live rather in open fields, while these two mice rather live in areas that are very crowded, in wooden areas. So one could argue that this high increase in UV sensitivity in the lower retina may have something to do with what happens in the upper visual field in this environment, but not so much in this environment. Recently, the distribution of the M and the S cones has been studied in more detail in the lab of Wei Li, and he found that it's even more extreme. There seems to be a specialization in the ventral retina that has a higher concentration of S cones, and this points towards something interesting happening in UV range in the upper visual field. This gradient also has, is expected to have an influence on color vision, of course. There are behavioral studies that study color vision in mice. There's an older study from the Jacobs lab where it showed that mice can indeed discriminate colors, and a more recent study that shows that mice can discriminate colors, but they're much better when the colors appear in their upper visual field. Okay, starting from this, we wanted to capture the natural environment of mice. We wanted to see what's happening in different areas of the visual field, and there are different ways to do this. There is so we want to capture the scene here. This is looking from a mouse scene in the field, and you want to see what's happening up here, what's happening down here. And of course, you want to do this at the spectral bands that are relevant for mice, so UV and green. So to do this, there are different approaches. Recently, there has been a study published from the Nielsen lab where they used a very nice approach to characterize visual scenes at different daytimes in different environments, at different weather conditions, and they generate these plots here that summarize a natural environment, visual natural environment at different positions. But this approach relies on static scenes being collected over a day or even weeks or so. And we wanted to use a different approach. We wanted to record movies from the natural environment. These movies might not be ideal for statistical analysis when you're just interested in chromatic properties, but we also wanted to be able to use them as a stimuli for retinal experiments. And I will talk about these experiments at the very end of my talk. Okay, so what we did was we built a camera on a stick, so to say, and this is the student who built this camera, Yongrong Q, together with his colleague Cici, and they put this camera together, and then they walked through the woods where we know that mice exist, and to the fields and the recorded scenes. So this camera is held on a gimbal, which keeps the lens here as a fisheye lens parallel to the ground. And this construction allowed us to keep this very close at a level where mice actually would sit and look. The camera consisted actually out of two camera chips. So these are two Raspberry Pi cameras that have been prepared for, one is more sensitive to UV light through a special preparation, and they sit behind bandpass filters so that one pathway is responsible for the UV and one pathway is responsible for the green. So by using bandpass filters, we could make sure that we capture the spectral ranges that are interesting for mice. And by using a fisheye lens, we also approached the visual field of view of mice. So there's one thing that one has to consider when you record scenes from the viewpoint of a mouse, and this is of course eye movements. And in the last couple of years, there have been a lot of studies about eye movements in mice, and they make very interesting things with their eyes. For example, if you look at this talk that I recommended to you a couple of slides earlier, it shows that mice can have this saccade and fixate pattern when they hunt, for example. So they use their eyes actively together with their head to fixate. But during long times when they just roam around, they also use their eye movements to compensate for head and body motion. So they keep the retina the horizon still on the retina. So we assume that our movies would capture more these positions. So by using the gimbal, we stabilize our camera to the horizon in our movies. So just keep this in mind. The movies, of course, don't contain natural eye movements of mice, but rather capture the scenes. So this is an example movie that we took here. This is the UV channel, this is the green channel, and this is the overlay. This is moving the camera close to the ground in a scene where there's some gravel on the ground. And so we try to move them along paths where mice walk, but of course, this is all the approximation by the person leading the camera. And I'll show you a couple of movies later again. So we took these movies in different environments. We calibrated the camera as well. So we have these two channels defined by bandpass filters, and the calibration worked by taking scenes in the natural environment and then taking images of LEDs of defined brightness in these bands here. And with these information, we could then define the dependency of a camera pixel here as normalized intensity to UV power or green power in the images. So we had an absolute measurement of brightness in these channels as well. And with this calibration, we could then take the raw image and calibrate it, so correct it for this relationship here. And this shows the readout of the camera calibrated and the readout of a spectrometer that is calibrated and took the same scene here. And if I gamma correct it just so that you can see better the differences, you can see that the spectrometer and the corrected mouse camera image are quite similar in terms of intensities. So we think that our intensity correction is giving a good approximation of the actual intensities in the scene.
Thomas, is it okay if I ask a question?
Sure, yeah. Go ahead.
I want to ask about the wavelength, because you said that, you know, if you just see a color TV wouldn't match, they won't be able to see, but also it will not elicit, I mean, it won't capture or activate the S-cones. So here the camera was able to record information that the human eye is not able to see?
Yes.
And
yeah, this is.
And also to broadcast it, I mean, later on to the mice, I assume?
Yeah.
Okay.
So I come to so we have a special visual stimulator that I show very briefly later. In terms of the camera, there's a very nice paper that uses these very cheap cameras for detecting actually smoke in the air, and that has a certain when you take UV images, you see smoke from plants and so on, from companies. And they just scratch off the filter layer from the chip because the chip itself is UV sensitive. The chip itself has a high spectral range. So we scratch off this layer from the chip and let the UV band just be defined by the filter. So the camera is actually UV sensitive.
Okay, thank you.
But that's a good question, sorry. Yeah.
No, it's fascinating.
Okay, so we took different scenes, like here close to trees or close to the forest, but these were all taken during the day in this case. Yeah. And then we wanted to analyze what's happening in the different parts of the visual field. So we divided these scenes up into an upper crop of the field and a lower visual field crop and analyzed these separately. And this here shows the correlation between the green channel and UV channel for the upper and for the lower visual field. And already we see here that while in the lower visual field, there's a very nice correlation between UV and green, in the upper visual field, this is not the case. So there's a much more, it's much more uncorrelated, the light in the upper visual field. We were thinking about classifying the scenes also by content, because scene content could be an important information, but this was very difficult. So we decided to divide the scenes by their mean brightness. So we defined 3 intensity groups: low intensity scenes, high intensity, medium intensity scenes, and high intensity scenes, and analyzed them separately. And in a way, this divided the scenes also a bit by content. You see these kind of scenes usually had a very high intensity, but if you had forest nearby, the intensity reduced. So close to forest scenes are rather in these groups here. And we took in total 1,500 crops from 15 movies. These images here have about 53 degrees of visual field. Okay, and then we looked into contrast. We used 2 different measures. We used the RMS contrast as a measure where we just apply a filter of different size to the scene and then calculate the contrast from this image patch. So we took scenes from the upper visual field, lower visual field. Here's the UV channel. This is the green channel. Here's the overlay. This is here for a kernel size of 2 degrees. 2 degrees on the mouse retina are approximately 60 microns. So that's a little bit smaller than the smallest ganglion cell receptor field. And then we can plot here the contrast in the UV and the green channel, and here the difference between the 2 channels. And we can do this also, we did this also for larger kernel sizes. So this is 10 degrees here, which would be around 300 micrometer on the retina. And what you can see from these scenes already is that the contrast in the upper visual field is much higher than in the lower visual field, and especially in the UV channel here. You can see this also from the histograms here. So this is the histogram of the RMS contrast for the UV and for the green for these 10 degree kernel sizes. And you can see that the contrast has higher values here, and it's shifted from the green contrast distribution, especially in the upper visual field, and much less in the lower visual field. And you can see here that this also depends a little bit on the scene. So this upper scene has a higher shift than the lower scene. We looked at this systematically in the 3 intensity groups. So this is again the histogram of RMS for UV and green. And here is it for the medium and high intensity groups. And what you can see here is that at low and medium intensity, you see this difference between the UV and the green distribution, and this disappears almost disappears towards the high intensity groups. And you can do the same thing then with a larger receptor field size. Here you see that this shift then gets more pronounced compared to the smaller kernel sizes. So this here shows the summary of this contrast analysis. This is plotting the median of this RMS contribution for UV and green for the upper and lower visual field as a function of kernel size. You see here with increasing kernel size, the contrast in the UV channel, especially in the upper visual field, is increasing, while in the lower visual field, it's not so big. And the difference between the 2 channels here in the upper visual field is much higher than in the lower visual field. And we see a trend that this disappears towards higher intensities. We use also a different measure for difference in distributions. This is just the median here, and the median is not describing the skewed distribution very well, but we can use also Jensen divergence as a measure of how different 2 distributions are independent of their shape. And we find here for the upper visual field, the distributions in the UV and the green channel are much more different than in the lower visual field. And it holds also to a certain degree for medium size for medium intensities and breaks down for the high intensities. So we can summarize from this, the RMS contrast is highest in the upper visual field and especially in the UV channel, and it seems to increase with the receptor field size to a certain degree. And the UV and the green channel are less correlated in the upper visual field, which indicates that there's chromatic contrast in the upper visual field. And this resonates very nicely with an earlier study from Katrin Franke's group, where they looked at different stages of color processing in the retina, among these also the ganglion cells, and they found several types of ganglion cells that show color opponency, especially in the lower retina. So the lower retina looks up to the sky. So this is the area of the retina where you would expect color opponent cells in the view of these chromatic contrast differences in the upper visual field. And this fits also to the behavioral study that I mentioned earlier, where they found that mice can discriminate UV and green to a certain degree better in the upper visual field compared to the lower visual field. Okay, so the second measure for contrast that we used was on-off contrast, where we use a centered around kernel of different sizes, run it over the field, and then calculate the difference between the on and the off components of this kernel. And also here, so this is again the same layout here. These are the channels. This is now for 2 degrees kernels. We see here that also this on-off contrast is higher in the upper visual field compared to the lower visual field. And this also increases with kernel size to a certain degree. These contrast distributions look a bit different because the contrast can go positive and negative. So we see here the distribution of contrast for the green and UV channel. And you can see that for some scenes, the UV channel is actually moving away from the green channel, so it's becoming negative, which means that in the UV channel there seems to be an overabundance in some images of dark contrast. And in the lower visual field, this is not the case. Here the distributions seem to fit much better to each other. So these are the histograms, for example. So how does this look on the population level? So here are the histograms for the 3 intensity groups. For the upper, for the lower visual field. And you can see here that at least for the lower and the medium intensity groups, there is this skew, the distributions are skewed towards dark contrasts here. You see this in the UV, but you'll see this also to a certain degree in the green channel, while the skewness is absent in the lower visual field. This is the summary data here, again, as the medium of this contrast distribution and the JSD, so the difference in the distributions as a function of kernel size. And we can see here that this bias towards dark contrasts increases with kernel size for the upper visual field, but not so much for the lower visual field. And here again, the UV channel is interesting because this bias towards dark contrast is more apparent in the UV channel. And this is something that has been observed in other studies of the natural environment, for example, in RedLive, where this was suggested in the retina, but there are also studies at higher levels of the visual system where this dark bias can be detected under some conditions. And this also relates to something that we did a couple of years ago. We looked at mouse scenes with a different method and looked at the distribution of contrast. So this is from this older study where we found that there's this shift of the spectrum of contrasts towards the distribution of contrast towards dark contrasts. And at this time, we looked at the properties of photoreceptors in the mouse. And what we found is that the photoreceptors actually seem to be already adapted to this kind of distribution shift. So while in the ventral retina, in the dorsal retina, we found that these M cones show a symmetric behavior when you excite them with dark or bright flashes here. So they respond to both, quite balanced in their response. The S cones here in the lower, in the ventral retina, they seem to prefer dark flashes. So they seem to be tuned to this shift in the contrast distribution. This seems to happen already at the photoreceptor level. And then, encouraged by this finding, we also looked into an older data set of ganglion cells that we published a couple of years ago, and looked whether we can detect there also a shift towards preference for dark contrasts in the ganglion cell responses. And so the data, so this is plotting the data from here just as a different representation. So this is the distribution of on-off contrast for different receptor field sizes. And this is the corresponding plot, but now every dot is a ganglion cell from this study here. And you can also see here in the ganglion cell distribution that there seem to be a number of ganglion cells that have a preference for dark contrast if you test them with an on-off stimulus. This is showing the on cells only and the off cells only. So the off cells seem to have a preference for dark contrasts. And this preference seems to also change with the receptor field size. So this shows these data binned by receptor field size, and there seem to be the cells with a larger receptor field seem to have a larger preference for dark contrasts. And this is something that the on cells show, the off cells show. Sorry. So the statistics here, so that for large kernel sizes, that in the upper visual field there is a bias towards dark contrast, seems to be reflected also at the ganglion cell level.
Thomas, can I ask you another question?
Sure.
Sorry for interrupting. Do you think the bias towards dark contrast is because the mice are looking at things from below? And if the lighting are usually, let's say the sky is bright, and so if, let's say, there are birds flying, etc., they would look, they won't be lit. I mean, they would seem dark from below. Do you think that's why I'm just thinking.
Yeah, so the shift and the dark shift, this is something that is still there's a lot of discussion about this. I get into fights with Tom Barden about this regularly. But so this would be a property of the environment. But what you're pointing out, it would be quite useful because any predator for the mouse would come from above and would rather be darker than the sky yeah. So this might be linked to predator detection in mice.
Thank you.
But I come to this also in a moment. Okay, so this was the statistical analysis of these movies, and then the student Yongrong, he was playing around with machine learning models, and he was thinking, could a machine learn, could a model learn this difference between the upper and the lower visual field, and could this explain why you have color opponent kernels in one part of the retina and not? And so he built this convolutional autoencoder model, which takes an input image from the natural scenes, applies a kernel that would be a ganglion cell or cell looking at this, and then tries to encode the features of the scene and reconstruct the image. And the difficulty is here that this model contains a bottleneck, so the information here in the middle is restricted through certain means, which means that the model needs to capture the important information in this latent layer here and has to reconstruct the image based on this reduced information. And the idea was, if we create a model that can reconstruct a scene still to a good degree, what would the spatial kernels look like that are needed to reconstruct the scene? Would they contain color opponent kernels? And so first we looked at how good the images that you put in are reconstructed. So this is here different regularization parameters that define how narrow this bottleneck in the middle is. And here you have input images, and these are just examples for the output of the reconstructed image. So here the output is quite good. Here it becomes a bit worse, but it's still good. And if you're reaching a certain level here, the images are not reconstructed very well. And then we picked this border here where the model is strongly constricted in its bottleneck, but still produces reasonable reconstructions of the scene. And we looked at these conditions. What are the spatial kernels that the model generates or learns when it's trained with upper visual scenes compared to when it's trained with lower visual scenes? And what we found here was that it happens more often that the model produces a color opponent kernel when it sees the upper visual scene images. And we tested this systematically with different regularizations and different runs. And consistently, the model, when it saw upper visual scenes, produced more color opponent kernels than when it learned the lower visual scenes, which we think is a nice illustration that the scene statistic might or could be able to influence what we see at the retina level in terms of color opponency. So the convolutional autoencoder trained with upper visual field scenes learns more often color opponent kernels. We tested this also with different kernel sizes. So in this case, the kernel size was 7 degrees, which would be a reasonably large ganglion cell. Interestingly, for smaller kernels, it doesn't work that well, but for larger kernels, it works as well. So there's a certain dependency on the kernel size for this. There have also been other ways of reconstructing or determining color opponent kernels from scenes, for example, PCA or CCA whitening, which is a PCA further development of PCA. So these are usual methods that have been suggested to decompose natural scenes into color channels. And of course, both of them also reproduce more color opponent kernels for upper visual scene, field scenes. So in this way, they are similar to our method. What we think is interesting is that our model here produces kernels that look physiological. So they look more center surround, while these other methods don't look naturally looking kernels. Okay, so far I've shown only scenes that have been recorded by during the day, but what happens if it gets darker? Does the UV channel is also useful when the sun is going down? So we were interested in this situation here. When the sun is at the horizon, there's this interesting feature of the spectral curve. So this is the wavelength, this is the power here, or irradiance. And when the sun is at the horizon, you see that the short wavelengths are actually increasing in intensity compared to the long wavelengths. And at this time, this might be an interesting situation for a mouse. Here, it might be useful to have a sensor for short wavelength, especially if you come out in the evening and there are predators waiting for you. So we took the camera out to at the sunset and the sunrise, and these are different parts of a movie that have been recorded while the sun was rising here. And this is the UV channel, and this is the green channel. And what was striking to us was that, and this is something that other people observed already, when you look at shorter wavelengths, the sky is much more evenly illuminated compared to longer wavelength. You see this in the UV channel. The UV channel, the sky brightens up here, while here in the green channel, you see the intensity mostly around the sun. So being able to have contrast vision here in this band here would be quite useful to detect a predator. This is quantified here in this graph here, where we plot the intensity along a line from the sun towards the sky. And for many of these conditions here, at many of the time points, the UV is actually a little bit higher than the green channel. So the UV illumination of the sky is more even at this time. And when we do our contrast analysis, we also find the UV channel is more interesting in terms of contrast. And there's also a certain dependency on the receptor field size. So then Yongrong and CC took a black drone out and took a movie of this just to show that it's nicely visible. And this is shown here. So here you see the black drone coming close to the camera, and you can see how well it shows up in UV channel, while in the green channel it's very difficult to see. So this is of course anecdotal evidence, but it shows that mice could actually make use of this channel to detect contrast in the sky during the day, but also when it gets darker.
Thomas, can I ask something?
Sure. Yep.
So if one of the purpose of this complicated machine of vision is to find the predator or something in the sky, so this would be something which is dark. And do you think it has to do anything with on versus off responses? If you are trying to find a dark object as opposed to a light object? And are there any evidences that this could be the case?
You mean that that the channels that are meant to detect predators are rather off channels?
Yeah.
There is, so I don't think that there's a specific ganglion cell type that's just the predator detection channel. But so I think it's rather a combination of ganglion cell channels that does this. It's possible that off cells play a more important role there, but not necessarily, I think. I think this is something that still needs to be tested and debated.
Okay.
Okay, so far we looked at intensities in these movies, and what we recently did, and this is partially the stuff comes from yesterday, is to translate this into what happens at the photoreceptor level. So how do these scenes look at the photoreceptor level in the mouse? And this is work in progress, as I said, and I'd like to walk you shortly through our procedure in terms of how to estimate photoisomerization rates in the photoreceptors from these scenes. So we want to have a function that translates camera intensities from these images into what happens directly in the photoreceptors. And this is our list, what we need to determine. And we did this, we generated a Jupyter notebook to calculate this. We will also publish this soon to get input on this. But I just want to show you how we think this could be done. So the first need is that we have to translate the camera pixels into some measure of power. And this is already what I presented you. We did this calibration of the camera. So we have the intensity of the camera as power in UV in the green channel. The second thing that was a bit more difficult to determine was how much light do we get through the camera to the chip. And because we have a lot of glass here, we have filters here, we have pinholes. So how much attenuation we have in this camera pathway. And for this, we measured with a spectrometer just the sky, and then measured through the camera channel with a spectrometer in the place where the camera usually sits, the same sky. And you see here that we lose much more light in the UV channel than we lose here in the green channel. So we could determine the attenuation basically from the light source to the front end of the camera chip, front end of the camera lens. We need also the wavelength specific transmission of the mouse optics. And this we took from literature, and we also took the pupil area as a function of light level from the literature. We then calculated how much an S opsin would be excited by light going through this band, because our camera doesn't contain exactly opsin matched filters. They contain these band-based filters that are matching the opsins more or less, but not perfectly. So we have to account for the fact that we are not optimally exciting the UV cone and not optimally exciting the green cone. So we have to account for these for these excitations. And we also have to account for the fact that the green cone will be excited by the UV channel as well, because of this beta band in the spectral sensitivity. And then the last factor was the collection area of the outer segments of the photoreceptor. So how well are they catching light? Because we wanted to consider also rods. And we took this again from the literature. And with these data, we could then translate our images into photoisomerization images. And this is what it looks like. So these are the camera scenes, this tree, UV green, this is the ground here. And now we flip the images because the dorsal retina is looking at this scene and the ventral retina is looking at this scene. And this shows how the images would look like if you were an S cone, an M cone, or a rod. We think that the relationship between the photoisomerization rates are fine. The level of photoisomerization seemed to be a little bit high for us, but this is something that we have to still deal with it. So just look at the images to that point. So now we can take also into account that the M cones here in the ventral retina, they have a lot of S-opsin. So you see here, this is how an M cone looks at this image here, but seeing both green and UV, having both an S and an M opsin. And you see that the contrast in this channel is a bit better than if you just use the M opsin, or if you are a rod. Now we can change the scene depending on the content of the co-expression in the ventral cones. So if you are really an M cone in the ventral retina, you would see this image here in this way here. So you wouldn't have a lot of contrast in the image. And if you increase the co-expression level of S-opsin, you reach, of course, a much better contrast, because now you're using this UV band and the contrast in the UV band. So the co-expression may simply be making more cones sensitive to this very contrast-rich information or in the upper visual field. So now we can take this translation of brightness into photoisomerization and color our movies that way. So this here is showing the original movie, UV green, and the overlay. And here is the S cone, M cone, and the rod. And then here are different overlays. And you can see here in the overlay, if you compare S and M with this co-expression ratio in the ventral retina, you lose all the color information up here, because here you're just looking with the S cones. You have only color information down here, if you're just considering the cone photoreceptors. However, if you are taking the rods into account, and mice seem to do color vision by comparing UV cones with rods, then you can gain all the color information back that you have here in the original movie. And we also try to illustrate, this is a different scene here, more in a forest. And you can see here in the UV channel, you have very nice contrast towards the sky. So you also illuminate clouds in this channel. You see a very nice contrast image. While here in the channel, when you compare S and rods, you get color information back here and across the whole image. This is the attempt of illustrating this, but this came last night at 11 o'clock and we didn't have time to optimize this. But the point would be here, you don't get very contrast in the upper visual field if you use S and M cones only, because of this co-expression. This is not unexpected, I guess. Okay, so do I have still a few minutes, or?
Yeah.
Okay, 5 minutes. So at the beginning, I said we couldn't use still images for this. We wanted to use the natural movies also as visual stimuli, and I just want to give you a glimpse on what we're doing with this. This is referring to your question earlier. So we built a stimulator that can produce UV and green images that match the spectral sensitivities of mice. This is described here in these publications. And then we can project the natural movies onto our retina, the isolated retina that we record under a 2-photon microscope. And this shows how the arrangement is. So this is the size of the movie. And this would be the recording area. And this magnification shows here ganglion cells at the level of the ganglion cell layer that we record at the same time using calcium imaging. And this is work done by Claudia from Katrin Franke's group. So if you then present these movies to ganglion cells and record the activity, it looks like this. So this is a movie here and the recording field recorded in the dorsal retina, and this is recorded in the ventral retina. So every dot is a ganglion cell, and every blink is, when it blinks, it means the activity increases or decreases. So we can read out what this ganglion cell mosaic does when it sees the natural scene that we are presenting. And so we use this data currently mostly to train a DNN model. So this is work by Lara Höfling and Christian Behrens. Christian is from Philipp Behrens' group. And they set up this convolutional neural network. And I don't want to go into details here, but the idea is that we can train it with the natural movies and then do in silico experiments on this model to test stimuli that we haven't shown to the cells in order to inform experiments that we can do later. And I will show you an example for this in a moment. So this DNN model is trained in the natural movies, and we have so this is the movie output of this model, the model output for this cell. This is the data for the cell. So we record the cell also with more synthetic stimuli like moving bars and chirps, so that we can recognize the cell type. This is here the response to the movie. This is the intensity of the green and the UV channel in the stimulus. And in black, you see the actual response of the cell, and in red, you see the prediction of the model, how this specific cell would respond to the movie. And then with the model, we can predict what would be the optimal stimulus for this cell. Here, this is the most exciting image that the cell would prefer. And we can calculate where it sits in the visual field and how big it is. And we can do this. These are dorsal examples, the ventral examples, and you can see we get different types of cells here, and also these predicted receptor fields differ depending on the type. So with this model, we can now study the properties of these cells also with stimuli that we haven't shown. And I just want to give you an example that links to what I told you at the beginning, to this regionalization of the retina on alpha cells, where you have the small cells here in the temporal retina and the big cells in the nasal retina. And our model is trained with data that was recorded across the whole retina. So we have cells that were recorded in the temporal retina of this type, and some that have been recorded in nasal retina. You see here the responses to these synthetic stimuli are very similar, although the cells are the same type but sit in different parts of the retina. These are the predictions of the model. And now we can test, for example, the prediction that these cells here should have smaller receptor fields because they have also smaller dendritic arbors. So we can present to the model stimuli like a spot that increases in size and measure the responses of the model. And this is what the model for this cell produces here. So the darker the spot, the darker the line, the larger is the spot. You see that for small spots, the response is high, and then the response is suppressed the larger the spots become. So this indicates that these cells have a small receptor field, while the cells in nasal retina that are large are not so much suppressed by larger spots. And this is then compared here in this plot. So this is the spot response as a function of diameter for these temporal cells. For the small cells, this is for the large cells. And you see that the model, although this model hasn't seen the stimulus, it's just trained with a natural movie, it can reproduce this, or it can predict that these cells should have a smaller receptor field and these cells should have a larger receptor field. And with this, we can now go back to the real retina, record cells, and test these kind of stimuli. So this is where we want to go with these natural movies in the first place. Okay, let me summarize. So I've shown you, we built this scene camera for recording UV green footage in the mouse natural environment. We want to use this as natural stimuli for building models and for testing the retina. We analyzed these stimuli, these movies statistically, looked at the contrast statistics, and we find that the upper visual field is quite different from the lower visual field. There's a lot of chromatic contrast in the upper visual field, which the mice may use, for example, to detect predators. And this seems to work also when it gets darker. That means at dusk and dawn. And I showed you at the very beginning that we are building these CNN models that will hopefully allow us to do in silico experiments that we can drive our experimental predictions with. Before I end, I want to mention one initiative that we are currently starting together with Tom Barden and the new postdoc in his lab. So we would like to have scenes, spectral scenes from many environments, also at different light levels. And we started to build a very low-cost spectral scanner that bases on this chip here that's only about 200 euros, and the rest is, I mean, maybe 50 euros. So with this equipment, you could build a spectral scanner that scans a visual scene at different times of the day and produces a spectral image. And with these images, we could then predict what certain animals would see in the scene. And we built this as a kind of telescope, so we expect that this also works at low light levels. So the idea of this project is also to look for color vision in the dark. We're developing this, and if you're interested in looking into this, then just contact me. Okay, with this, I'd like to thank you for listening and for the questions. I think I mentioned all the participants, I think, already. So this is in collaboration with Katrin's lab, Laura's lab, Philip's lab, Fabian's lab, Alex's lab, and this is a lot of fun. So thanks a lot, and if you have questions, you're welcome.
Okay, thank you, Thomas. So let's all make some. Thank you, and we have time for a few questions. Anyone? So maybe I would like to ask something. What do you think? Anything of this similar effects to be found in human? Some differences, not central versus periphery, upper, lower, temporal, nasal, large scale differences, or in primate?
Yeah, I think the primate retina is, I mean, is really very strongly determined by the foveal peripheral organization. So I don't expect, I mean, I haven't heard of anything that would further specialize in the periphery, but please correct me if you know it better. But the difference between the fovea and the periphery are quite big already, and it's not just the size of the cells, it's also the temporal properties of the responses. So I think this is the major regionalization in the primate retina, I would say. But if there's anyone else who knows this better, please correct me.
Yeah.
Hi, Thomas. This is Petri. Yeah, hi. Very nice talk. So I have a quick question, sort of at the end part of your talk, where you described the model and also the the cameras recording across different light levels. So the first question is that, have you tried to apply the model across different light levels, or are they is it done on a static light level, and sort of following up on that in that direction, that how would you kind of build a model across wide range of light? What would be the adaptation model to kind of put in there, or is the network sort of doing that automatically? I find it kind of fascinating how to build in dynamic range into models like that.
Yeah, so thanks. That's a very good question. So the project that I just presented at the end, this is first collecting the data. So we're not thinking about the adaptational aspects of this in terms of the model, but we observe. So we're currently looking into the point, how much is the model dependent on the light levels that we used for the experiments. This was also partially a motivation of this whole translating the images into photoisomerizations, to know where are we actually in our experiment, because we have an isolated retina, there's no optical apparatus, we blow it with light, and is this realistic? Yeah. So this is an attempt to get there, and this is indeed still an open question, whether we could use the model also to extrapolate across light levels if the model hasn't seen this. I would doubt this, but I mean, I would doubt this. So the question is, can we train the model with different recordings at different light levels and then interpolate over more light levels? That's an interesting question. That's actually something I wanted to talk to you later when we meet.
Okay, thanks. Yeah, I will do that.
Thank you.
Thomas, there's also a question in the chat that was written directly to me, but it relates to the issue of, wait a minute, what is the, why do the mice see UV? So you did mention that it may allow them to see different things, or see, for example, the upper visual field in a more uniform kind of manner without the disruption of, let's say, the sun, or, but are there different or additional benefits that you can think about?
I still think it's surprising that mice see UV, because I mean, UV light has more energy, and it's more dangerous for the tissue. So you would expect also more photo damage by UV light. And you could argue mice can deal with this because their lifetime is rather short. So if they collect a couple of problems on the retina, it doesn't matter so much. And this could also be the reason why most more long-living animals have a UV screen in their lens or in their cornea, so that they have no UV sensitivity in that sense. And so I'm convinced that by having a short wavelength sensitive band, you can extract more contrast information from the sky, and this might be enough to together with the fact that it doesn't hurt with that lifetime to have a UV sensor, that this should be enough, or it should be an advantage in evolution. But if you're interested in the evolution of photoreceptor pigments, then you should listen to Tom Barnes' talk on Friday. He's thinking about this quite a lot, and he may have a different explanation why mice have UV cones, more from the evolutionary point of view.
Okay, thank you.
Okay, thank you. So any other questions?
Yeah, hi. Regarding the spectral opponency in your naturalistic videos that you collected, I was wondering, you saw like the differences in the spectral opponency for the different light intensity levels that you had in different videos that were recorded. And you made this remark, I think, that it might be related to them being like closer to the woods also. So I was wondering, what do you think is the explanation for the spectral, for the difference in the spectral opponency that you see at different light levels in your videos? Is that like really just related that you have like more trees and such, so basically that the upper visual field becomes more like the lower visual field in the darker environments? That's all altered, or is there like something else that you've been thinking about?
Yeah, I think it's related to if the sky is open, then the scene gets much brighter under the conditions we recorded, and then you typically don't have a lot of things in front of the sky, so you're losing these structures that produce contrast. I think this is partially the reason why this difference breaks down in the for the high intensity scenes. We looked into whether it could be related to saturation in the camera, because the camera is of course limited in its dynamic range, but it doesn't seem to be the case. So I think it's rather related to what is the content of the scene, that the content of the scene determines the mean intensity, like you said, edge of forest versus somewhere in the field.
Okay, thanks.
Okay. So if we don't have any other questions, we can thank again Thomas.
Thank you for the invitation.
Thank you. This was a really fascinating work, and we are looking forward to see some more results from this coming in the next year.
Me
so
too.
Thank you all for being with us, and see you next week for other talks.
Well, I want to make some, I'd like to make some announcements. That next week Israel is going to additional elections, so we have we won't have a talk, and the week after we've got holidays, so we'll be back on April 6th with Leon Duell. So I hope everybody will join us then. We'll send also email notifications about these, but yeah, thanks again.
Thank you very much.
Wonderful talk.
Thank you.